The culture facility can help answer key questions within larval ecology, biofouling, and evolutionary biology. More specifically, it can be used in studies on receptor-ligand interactions during larval settlement, development of new antifouling technologies, and the evolution and function of genes involved in osmoregulation.
The main advantage of this model organism is that the barnacle Balanus improvisus is robust, reproduces all year round, and has a relatively short generation time. Though this method can provide insight into the culturing of barnacles, it can also be applied to the culturing of other marine organisms. Generally, individuals new to this method will struggle because of the large number of details that are important for success, such as the supply of good seawater, robust production of high-quality feed, and a constant attendance to cleaning and feeding the cultures.
Demonstrating the procedure will be Martin Ogemark and Anna-Lisa Wrange, technician and researcher working with barnacles.
To begin, set up frames that can hold several transparent thermoplastic panels. Drill 2 holes in the top corners of each panel and use cable ties to attach the panels to the frames. To collect adult barnacles, deploy the panels vertically at a depth of 1 to 3 meters in calm waters. After allowing barnacles to settle and grow to 5 millimeters in diameter, bring the panels into the laboratory. Then place the panels vertically in a stand with milled grooves in a polyethylene tray. First, using non-toxic water-resistant tape,